right? So, yeah. Just once again, reminding you of my name, Anton, and I'm from Sean's lab at NCBS. All right. So next, I would go on to present small introduction to. Okay. Just okay. So I'd like to present a small introduction to the system of study that I do. So when you look at isogenic cancer cells, these are cells that are genetically identical. They still, however, bear many heterogeneities or develop many heterogeneities which are of non-genetic origin that can arise either due to epigenetic modifications, gene or protein expression variations, and post-translation modifications, etc. Now, these heterogeneities, which are present in isogenic cells, are particularly highlighted when we look at, when we expose these cells to a particular drug, chemotherapeutic drug such as cisplatin. Exposed to a drug, we see that not all of the cells die, and there are certain cells that still survive the treatment, leading to only a fraction of cells dying in the population that was actually exposed to the drug. This phenomenon is called as fractional killing, and it provides certain challenges to understanding how the population dynamics of a cell or population of a cell culture can be predicted from single cell attributes. Now, here I present a graph which is from data provided to us by our collaborator Adrian. In this graph, this is a semi-log. This is a log versus log linear plot of population versus time of U2OS cancer cells. This has been divided into two sections: one that is before addition of the drug, and one that's after addition of the drug. The drug here is cisplatin. And as you can see, that most of this graph is behaving in a linear manner. It shows a linear relation between log of the number of cells versus time, and thus is an exponential growth of population of cells, except for this small region where it is this transient period. Now, the general or the popular models currently used to find out or relate the population growth rate, right, to calculate the population growth rate from single cell parameters is the exponential model, which takes into account that the effective growth rate or decay rate of a population, which is mentioned here as K, is given as the difference between the birth rate and the death rate. And the birth rate is nothing but the inverse of the mean division times, and death rate is calculated as an inverse of the mean of the death times. And the difference of the two is usually known as the effective growth rate. Now, as one would see that with increasing concentration of the drug, right, going from low to medium to high, and this inequality is followed by the effect of the effective growth rate. And this is generally explained either by saying that the birth rate has reduced or the death rate has increased, which in turn results into such an inequality, right? Now, having got both the population as well as the single cell data, we thought to ask a very simple question. How well, with this model, can we explain the data that we have got, right? The population graph for which I've shown here. And how well can the current models predict these slopes, the K3, K2, K1? Now, a little about the data. This data is single cell obtained from single cell time-lapse imaging, which basically provides tracking information. It tracks the lineages of the cells, thus provides the entire lineage information of the cell. And along with it, it also provides us the single cell times, which can be calculated. Now, then we get certain distributions of the intermitosis as well as the apoptosis times, and then correspondingly we can obtain the birth and the growth death rates. Okay, so this is the result that we got for the before the drug period. Before the drug period, here, if you look at the graph on the left, this has a linear fit, which is nothing but directly what the slope of the experimental value is. Then there's the line in blue and red, which is the von Foerster as well as the inverse of the mean methods. Right. So for the data set that was before the drug, we estimated this population growth rate from single cell times using two methods. One was the inverse of the mean, and the other is the von Foister's method, which provides us with an equation of an Euler-Lotka that estimates the population growth rate. Now, interestingly, with this distribution in mind, right, of the intermitosis time, when we calculated the population growth rate from just the inverse of the mean, we found that this deviated by about 73%. This red graph over here, this red line with the black, the difference came to be about 73%. And this wasn't pretty surprising because anyway, this distribution of the division times that we have is pretty broad. So it is expected that a mean wouldn't be good enough to cover the entire distribution or to represent the entire distribution. We then use the von Foister method, which allows us to obtain the estimate from this entire distribution. And basically given a certain weight to all these different division times, how do they contribute to the population growth rate, right? So that also provided us with an 22% error. And this was a little surprising because, I mean, this is just a model for, this is a well worked out model and a pretty popular model and condition which is, for which it has been used. This is without any drug. But then I was pointed out by my TAC members that there were certain oscillations here in the graph, and those I had no clue what they were. So to figure out, okay, what are the estimates of error that I should get in this using the von Foister model, I did a simulation wherein I took intermitosis time from the experiment and creating these lineages of cells, obtained both the population time curve as well as the intermitosis time distributions. These were then, these intermitosis time distributions were then plugged into the formula to obtain the estimate for the population growth rate, right? So, and doing this all 1,000 times to know what the error limits are. And interestingly, what these oscillations led to was a phenomenon of synchronicity in the cells. How relatively, how similarly aged are the cells at the time of seeding, right? And this synchronicity had a very strong impact on the error that was obtained using the estimate of, or the deviation in the estimate from von Foister to the actual population growth rate, right? Which is nothing but the regression fit of the experimental value. And what we saw was this curve that there were for the error rate of around 22%, which is for our data, it corresponded to synchronicity value between 50 to 75%. Now we are looking back at the images to find out what the synchronicity was actually in the data that we have, and correspondingly calculate the estimate of the population growth rate from just the three data point, from just the three attributes that we took of the single cell data, right? That is, okay. So given the initial age and synchronicity, we can figure out what the single cell division times are. So given the initial age, synchronicity, and the single cell division times, we can determine the growth rate of the population. So this is what we got for the case before the drug. Now, after the addition of the drug, first of all, we were surprised to see that both the distributions for the apoptosis time as well as for the intermitosis time, for all the three different concentrations of the drug, remained nearly identical, right? And but this was not so for the population decay rate, right? You can see that they are very different, very distinct slopes. So this surely, I mean, as expected, we were not being able to calculate or estimate with a good accuracy the population growth rate from single cell division and death times. So when plugged into the formula for von Foister, as well as for the inverse of the mean, right? And looking at the difference of the rates of the birth and death, in case of the inverse of the mean, we found that the deviation was pretty high. And all the models, both the estimates, were giving positive values rather than when the decay rate was actually negative, right? when the population was actually decaying. So then we questioned that, okay, gave a little thought that when we are looking at these intermitosis times as well as the apoptosis times post the experiment, we are missing one point. Thank you. We are missing one point, that a fact that a cell post introduction of the drug has either is to divide, right? Happens to divide, or to just stay alive, or it can die, right? And when we would be looking at a cell that has died, right? If it could have also divided, given that it was given a sufficient time, right? So what I mean is that when we look at the population, when we look at the distribution of apoptosis times, there would be many cells in that distribution that could have also divided had they not chosen this fate, right? So now what would have been the actual distributions from which these cells were choosing these fates? And to calculate that, and there must have been, this problem can be compared to a competing risk problem, where there are multiple fates that a cell can undergo, and but it chooses only one, and what we get is only the final representation, only the final result, and the distributions are thus in a way biased. So to remove that bias and to figure out what the original distributions were from which these cells chose a probability to divide or die, we did using an MCMC, and finding, first of all, calculated what the likelihoods would be, and used MCMCs to sample from what distribution these cells should have chosen, either to divide or die. Now, these were the distributions that we got. And so this is for the intermitosis time post-inference and the apoptosis time post-inference. And so here again, what we saw was that the apoptosis time did not change much, whereas the intermitosis time showed a deviation. And this is for cisplatin, which is a cytotoxic drug. All right, so at least with this changing distribution, we thought, does this provide any better result in the model? But again, we saw that there was a still high deviation, and the best that we could achieve was for the high cisplatin dose, where there was an error of around 44% using von Foister model, right? So this leads to a question that, okay, what are we missing in this entire method of estimation? Are the single cell times enough, or is there something else that we need to incorporate? So looking back at the previous work and some literature already published, here we looked, we found, it has been found that the sisters in a population are known to exhibit similar fates, and there is a very high correlation in their fates that has been observed. So can this information of lineage help in provide, estimating a better, estimating the population growth rate from single cell data, that is including this lineage and attribute into the estimation of population growth rate from single cell information? This is just a simulation run, and maybe there could be switching between different states that eventually lead to have a higher chance of dying or dividing. All right. This is just a summary, and thank you.
So we have time for one question. Yeah, can you please use the microphone there?
Hey, Anton, great talk. So I had this just thought, like you mentioned about drug concentration, but did you also mention about like cellular density that you, my point of the question is like, depending on the cellular density, sometimes the behavior of cell also changes, right? So what I'm interested in, if you have the same kind of growth rate, death rate, depending on the initial cell density, does that matter? Like if there is any crosstalk between the cells and stuff like that?
So yeah, these definitely would matter, but however, in this experimental data that we have got, these factors are not playing a major role because these population is pretty scant, and even if you look at the population density, like the population growth curves, they are not as many that would start that would start affecting or bringing about these concerns. So does this,
But when you let the cell grow, right, they're going to make small islands. So locally, their density might be different at some point, right?
locally their densities might be different, right? And mhm. All right. Thanks, Tapa. Maybe I'll think about it. I mean, I'm not very clear. Yeah.
Let's thank once more.